Okay. We're going to move on to the lapis data set. As you might guess, this is a thin section of lapis. Creative naming, I know. Lapis lazuli. It's really cool. This is taken at 14.3. We have two lapis folders, and I can't remember which one is which, so we're just going to go with lapis 1. I think that'll work. Yeah, let's try that. So I'm going to start with the course data file here first. And we can hopefully take a look at it. Oh, there we go. Oh yeah, I remember this one. There we go. So that's a nice course data file. You can see there's some chunks of stuff distributed and probably some big honking hunks of pyrite in there too. Those really lovely intense sulfur spots. Lapis is cool because it has a really cool sulfur species in it. That's a trisulfur radical that makes it the cool ultramarine blue. So it has really good sulfur spectroscopy. So here's a map that was collected as a course map to give us the context of the entire thin section. And I did some chemical mapping, so we actually will slide in a little chemical mapping here just for everyone, to keep everyone happy. But of course, I could not map the entire thin section at high resolution at many energies, 1, 2, 3, 4, 5, 6 energies. That would have been prohibitive to anyone's beam time, even mine. So we see something like this bit here. That's this little corner, right? And if I look at that and I come back to my course area, you can see that that's this little guy right here, right? That little half moon corner there and such, right? And what I'd really like to be able to do with this data set maybe is have my high-res chemical map overlaid on my contextual sulfur map here to put in a cool figure in my paper, right? That would be kind of cool, right? So let's see if we can do that. Okay, so we're going to come to the lapis data here. And for chemical imaging, the first thing we need to do is import all those energies into the same data file, because they separate into different data files. So we're going to import data from, and even though we started with a low one, I like to re-import all of them over again, because when you do it this way, it puts the energy on them, and then I can actually think about what I'm looking at again. The window comes down in the bottom corner here that says what channel. The sulfur is the one that's varying. So I click on sulfur, and it gives us sulfur. And we see, oh, look, things change. Woohoo. Awesome. Got one bad line there that I'll need to worry about, but we'll make it work. So what we're going to do is I'm going to edge remove here. There we go. Get rid of the one bad pixel. And we can take this data, and we won't do a full analysis on it, but we'll do a quick little simplex volume minimization method that we talked about in the morning. So I'll say do PCA, brings up the PCA box here. I'm going to highlight all those sulfur channels here down. And the CIVM is right here, CIVM. Click on that one. Let's say 3 end members. Let's say 4, not /4, 4. And this is the way that you remove the noise. And so I don't want the noise to dominate my CIVM determination, so I'm going to give this about 1/4 of the mean is what I consider noise. We'll see if that works. Automatically, it's going to come up with a bunch of stuff, and it looks like this. So what did that do? It created a bunch of plot markers where it thought data was, and apparently I forgot to move the right corner off a little bit, so that looked horrible, and we'll redo this. And those are the spectra it picked up. But cool enough, it actually picked up a sulfate here, which is the first one here. and it picked up a pyrite and a lapis, it looks like. So, I mean, that's kind of what I was hoping it would do. Woohoo! Science. Yeah, actually looks pretty good, actually. Actually, not surprised. Not too shabby, not even with that kind of crappy. Let's see how these look. So there's our sulfate area. There's our pyrites. That's version 1, version 2. Yeah, it's okay. We can actually go with this. I'm actually okay with this. What I'm convincing about here is that, it doesn't look too bad. I was worried about these 1 and 3 points being so far off to the corner that maybe we either didn't make our threshold of noise too high enough, or that we had a feathering of incomplete lines on the outside. It looks like I don't need to worry about that. We could have tried to make it go higher concentration. Let's try that one more time. You'll notice now in the newer version of SMAK, I've tried to make these colors look a little bit better on the lines because I really had a hard time telling the third and the fourth one apart all the time. It got really, really annoying. But there's a way now you can save under View and the plotter color palette. There's several palettes. There's the ones that are a bunch of orangey ones, a bunch of grayy ones, some brown, blue, greens, accent, ones that are colorblind friendly palette. These are 2 ones that are, again, made to maximize these color contrasts. If you Google Husserl and Hulse, you'll see what they are. They go through and try to at least make things as different as possible so you can actually see the difference between the lines. Spectral is kind of the old-fashioned jet-y kind of way of going through things. This one's a pretty good one. I like it. We'll keep this on Husserl though. So you can change your color palettes. I'm not sure if it redraws it. Probably won't until next time, no. But I like this one here. So hopefully you can see your lines better. We are going to redo this real quick. I am going to say remove those data sets. Remove. I'm not going to bring this so you guys can see it, but I'm just going to get rid of those civums and redo it. There we go. I'm also going to clear all of these data points. There we go. And we're going to try to do this one more time. Nope, I don't want to make a tricolor plot. I want to do a PCA plot. PCA analysis. Here we go. Highlight all of these. Sieve them again. We'll still do 4. But now we'll try to make this half the mean rather than a quarter of the mean. Hopefully we can, actually, no, those, keep this at a 1 on the mean. Let's be hardcore. Oh yeah, and now all 4 of those are closer to the center. I like that a little bit better. So here's our sulfur map. There we go. Maybe slightly more relevant, maybe. Put it 1, 2, 3, 4. Yeah, actually, it looks pretty the same, but it's good to know that's not completely different. It just shows that you have a different extent of the species going out here between 2 and 3, and that is really the difference between the blue and the greenish pale one and the brown. So that's this monosulfide versus the all the way through the middle. Okay, beautiful. So say we had this great chemical information. Now we want to put that into our data map. One key part here, particularly for those of you who are on the PCs and you unzoom to see your plot markers, we're going to want to re-zoom in one more time here before we go to the next step. Okay, so I'm going to edge removal one more time just to be safe, right? Now, the biggest problem here is that these were collected at vastly different resolutions, right? So what were they collected at? Maybe you didn't take great notes like me. I never take notes anymore. I'm a bad person. If we go to process, is it process? Yes. I can say to change resolution, okay? If I just go to change resolution, this tells me, bring the window up here, what the data was collected at. And so this is telling me that this is collected at roughly 10 by 10 microns. And so if I don't do anything to change this, it will basically just resave the file. But this also resaves it in the current zoom. So if you find that edge removal really annoying, like I do all the time, and you want to save a file without that nasty edge, you can do your edge removal, change resolution, and save that data, keeping it the same, and you'll remove your edge for good into a new file. This is going to be important because we're going to import this file into the coarser one, and we don't want to have a big red line at the left side of our data. So I'm going to save this, even though it's not changing the resolution. I'm going to save it anyways. And so here we are. I usually am in the habit of getting rid of the energy in this case and just calling it a multiple energy region 1 new resolution. Okay. Do you want to load the new file? Why, yes, I do. Thank you. And here's our data. All nice and fixed. It's even so good, we can get rid of this one. We'll close that. And it's probably going to yell at me for wanting to discard it, but yes, we can do that. Now this guy, much bigger data set, much larger area. I'm sorry we're going to do this to people with small screens, but we're going to go to process here, and we're going to change resolution to this one and make that match. So this was done at 50 microns, right? We're going to change that to 10. And we're just going to straight out save that. Yes. So now we'll have two different data sets collected at the same time, same motor positions. Oops. Oh, yes, we do want to load that. And it's going to do a bicubic sampling to get us onto the new resolution. You can see we can't see the bottom of our screen whatsoever. And there we go. But here I could actually now go to view and go to my scaling factor and make this 4 times smaller. And there it goes. It looks just the same, really. Right? There's our data. I've basically upscaled the resolution to 10 microns. It's fit smoothie lines through it all. But now they're on their exact same resolution, right? And so now, if we're lucky, I'm going to import the multiple energy data from this file, into that one. Because now we're at the same resolution, we have the same energy areas, we can do that. The tricky part here is if you say import data from, which is the cute little button here, this is the automatic rapid import. It better be the same data size and the same step size. We don't have that case, so we need to go to the old school import, not the quote unquote rapid import, which is what the button does. So we're going to import data from the file. We are hopefully going to choose our multiple energy data here. We're going to open it. It's going to warn me that, you're sure you want to do this. They are different sizes. This is the whole point of what I'm trying to do. And it didn't yell at me too much now. It gives you a nice little warning again. And now we say, let's do that with all of our component data that we just PCA did. We're going to click OK. Oh, no, I'm sorry. Now it's asking me down here. Whoop, bring it up. Do you want to name it? Sure. Let's call it that. It's telling me that it's going to pad 14,000 pixels to the top, 1,400 pixels to the top, 200 at the bottom. It's actually looking at the coordinate positions of each file and trying to tell you where you should put it. And so because this is the same beamline, those coordinates are the same coordinates, and we're in the same scale, same 10 micron scale, we should be like, yeah, I want to do that. And I'm going to do that with all the other ones here too. They're off screen, but we'll deal with that and just say, okay, okay, okay. How many more of these do we have? Okay, okay, okay. And so now, boom, there they are. And we could even say, let's zoom in on this section in here. All right, there we go. We can even make it less viewy. So we're going to go back and make this 1x, maybe 3. There we go. That looks nice. And now we can see this idea in the context of, say, all the sulfur. And now it's a little bit coarser, but you see how that pyrite matches nicely, right? So you can see the whole context. And maybe you can go like, oh, how did that look with the silicon or the phosphorus on the big scale? You can always choose to go downscale rather than upscale, but I usually go to blurry or coarser stuff. Now you could even say, let's do a tricolor with this data. You could say, you know, make pyrites red. Not sure what pyrite color should be, but we'll call it red. Make our other sulfatey bits green. And let's put all the phosphorus background in blue. Oops, sorry, wrong color. Blue. And let's go back to green here. All right, or we could even say the full sulfur context. That should be this one. You know, I see all the other pyrites in there. We would have to really scale this down to see all the other features. There you go, for instance. This is another really good example of maybe we want to look at this in a different way rather than just RGB data sets. So RGBs are cool and all, but we can also go to what's famously known as the multifader. So here's the multifader panel. I use this mostly to combine things like photo images to X-ray data and that kind of stuff, but we'll use it in this example as well. We'll come back to this in a minute as well with our other data. But we're going to take that sulfur data and make it grayscale, as the example there shows. And we'll come down and take our pyrite data here and the standard gamma there. We can take a look at see what that looks like to start with. And bring this over here. And so here it is in grayscales here. I don't like that gray channel. Let's go with what sounds good. Do the Veritas here. Okay. It really just shows those pyrites again. You know what we'll do? Let me cancel this for a second. We're going to make, there's a couple ways of doing this. You can go to map math for this, but often you can also go to process and say, do a log transform. I do this a lot when I want to do lots of channels for log transforms. In this case, I'm only going to do the sulfur. There we go. Let's go back to the multifader, so it'll see that change in the data channels. We'll choose the log of the sulfur now. We'll do this. Looks a little better, maybe. I'm still going to get out of that grayscale stuff. Still kind of see it in there. We can also make this a stronger contribution. There we go. Now you can kind of see that in the background. And if we choose the second component here, maybe, you can see how that overlays on top of that now. And so doing multiple, rather than just doing red, green, blue overlays, you can do different color map overlays as well. And one problem we often have is that this isn't necessarily a dark background. And so I can bring up a lower threshold to say if it's 32% of the background, make it transparent. And now it's all transparent. Oops. Now it's not going to work on me anymore. That's too strong. A little bit of background there. In any case, you can play with how these come together. I think it's a little too, that's being a little too harsh on that. I'll have to take a look at that later. Anyways, we can combine data sets and now combine different color schemes on top of that as well. The multifader. I think we have the multiple resolutions set up pretty well there. Same beamline, same data sets. That worked out pretty well. Don't have to do anything too fancy with that. I'm going to do one more quick multifader example here. This is one that Joss has worked really hard on. So what we have in this case is some bright field data. This is actually a, let's take this off of log and put this into something that's a little bit more, well, Jet works for now. There's different optical scopes. That's the laser head here. And there's two fluorescent tags that were marked on this for different populations of bacteria in the sample. So this is fish that's been done. And here it's actually quite important to have the red and the green because those are the colors of the tags. And so we don't actually want to make jet color maps of the tag. We want to make a tricolor proper map of the red and green images on top of the bright field image and with some phosphorus data in the background, let's say. And this sounds like something that you'd really end up doing with PowerPoint or Photoshop at some point, but let's see if we can make it work in the multifader. So we're going to take our view and go to the multifader again. I'm going to make a quick note here that it's very important if you're going to use this particular method that anything that you want to do tricolor with, you start naming it red and green. Because I've been too lazy to figure out how to make a multiple section through this. So it's going to key through and find the first two things that start with red, green, or blue to make the tricolor map. So we're going to start here and go with the bright field data. And I'm going to add on some phosphorus data in standard gamma. Sure, why not? And we get a display like this, a little psychedelic. There we go. We see the camera image. I'm going to see if I can make the standard gamma with a lower threshold again, see if this works. There we go. It's bringing down some of that data so it's not all just blue on the background. And now you see the optical image in gray with the high points of the phosphorus here now in the color. So I've gotten rid of that blue background. That was kind of making it all blue look. I don't want it to be blue. I want it to be just the high points. So that's what this little threshold button here is doing here. There we go. Now we want to see those cool little bacteria. So I can add a new channel, adds a new bank here. And now we just say use tricolor. I have to click something on here. It doesn't matter what it is, but it is going to make a tricolor map from the red and the green channels. There's no blue, so there'll be no blue on there. I preview that, and of course it added that on there. It made it really dark because we haven't lowered the threshold. We again do that same trick. Boom, there you go. And now we have a tricolor map where you have the yellow and the green here, the microscope image in gray, and the phosphorus in this bright weather kind of map kind of thing. So again, ways of multi-fading all your data together. You can play with all the settings until you get ones that are just perfect. But again, tricolor maps, color maps for elemental data, and a microscope image in the background. So again, if you think about multimodal, I mean, you can keep adding things on here, and you're playing with those background thresholds. You could put phosphorus in one, sulfur in another, keep on building up your different channels. And this is also really important if you want to think about how do I display like 5 elements of data in a tricolor map without things being ridiculous. And so you can use these different oranges, reds, greens, purples, and blues and just set the thresholds so that you only show the top 50% of the data. So it only shows where you have the high data, and you have pastel-y oranges, blues, purples, greens. And you can kind of overlay different colors and make more than just your RGB. Cool. And again, we're all still always working for making cool artworks when it comes down to it, really. Another thing that I have done before, just to really drive reviewers crazy, I'm just going to draw this up here real quick, is, you saw a black color marker and a white one, right? But if we look at these markers, I'm just going to add, say, 3 here. I'm just going to put them in kind of random spots here because I don't want to be precise. I can do this, and I can now make these big circles, so they're a little bit more visible. And let's say I was doing a data fit, and I had my Zanes data, right? And say I took 3 Zanes points here, and I had one data point that was like 90% of one species, one that was 50% of another species, 50-50, another one was a 20-80 mix, right? I can think of now customizing my color of this, so that this color, let's not just go to this, let's go to this slider here, color code my markers to color code the percentage of my Zanes fit that I did on the full spectra later on, right? So this one here, let's say if I'm doing just a 2 plot, I'm going to do this by eye, but that's about, say, 80-20 here, right? Okay. This one here is going to be 0 here. I probably could type out the hex values correctly, but again, I just can't be bothered to do math in my head right now. That's that one. And then this one here, again, let's see, maybe this one actually had a little bit of the other one here. So let's do this one about that species, call it that one, and hopefully this math works out, but say call it that one. No, let's make it less. Oh, that's actually okay. Make it more blue. Half, quarter, quarter, right? And now you've color-coded your Zanes to your speciation plots that you did